Hi, guys. Let's talk about CRISPR. This article came out September 11th, 2020, from Common Health. CRISPR comes to COVID, a pandemic pivot, and the push for a simple coronavirus test. About 5 years ago, when the gene editing tool CRISPR was just starting to sweep labs around the world, a famously brilliant scientist encouraged two of his grad students just to play around and tinker with it. A lot of things that we failed to try, says one of those students, Jonathan Gutenberg. And that's okay because sometimes you find these things are really, really awesome, though they might not seem so awesome at first. CRISPR is often compared to a word processor that can hunt down the genetic equivalent of a specific word in a test and precisely slice it out. The grad students found that a particular molecular scissors, an enzyme called Cas13, would slice RNA, the chemical cousin of DNA, at the right spot, but then willy-nilly would go on to cut RNA all over the place. There was actually a time when we were like, should we even publish this, says Gutenberg's research partner, Omar. It's really weird. It really doesn't align with what we wanted for these enzymes, which is targeted cleavage, not cleave everything. But the scientist, Fen Zhang, a CRISPR pioneer and a leading researcher at the Broad Institute and MIT's McGovern Institute, saw potential importance there. He recognized that this is a unique feature that we should publish. It could be useful one day. We wanted to kind of hide it under the carpet because, this is not what we want. And months later, we realized that it could be used for detection. Those runaway scissors could be used for diagnostic tests. Once they're triggered to start cutting in the presence of, say, a specific germ, the continued cutting can also turn on fluorescent molecules that signal germ detected. They call the method Sherlock. Like CRISPR, it's an acronym. If you must know, it stands for Specific High-Sensitivity Enzymatic Reporter Unlocking. In 2018, the team helped found the company Sherlock Biosciences to start developing CRISPR diagnostic tests. A new use for CRISPR beyond its advances is brought in genetic diseases and research ranging from cancer to agriculture. Then this year, the pandemic hit. Now, with the coronavirus test in short supply around the country, CRISPR could help. Sherlock Biosciences is using the gene editing system in coronavirus tests approved in use in healthcare, the first CRISPR product to reach the market. The company hopes to come out with an at-home coronavirus test next year. Others are on the CRISPR coronavirus case as well. Like the World Series, the Sherlock company's push to develop a CRISPR-based coronavirus test felt like being in the World Series. It really did, says principal scientist Christine Cotticchia, because there was so much on the line every day. You plan so much, you train so much, and then it comes down to the day, and it comes down to the data. Adds Sherlock Biosciences CEO Rahul Donda to office laughter. For Christine, it felt like it was going to the World Series, and she was the pitcher. Donda says in normal times it would have taken the company at least another couple of years to bring a CRISPR test to market. But in February, with the pandemic looming, he appealed to the board to let Sherlock Biosciences shift nearly overnight to develop a coronavirus test as quickly as possible. Really, back in February, wasn't this all announced here in March? Wow. And each of them, to their credit, responded by saying, this is the mission of Sherlock, to solve these problems, he says. When Donda told the Sherlock staff, he saw them all turn on their chairs simultaneously toward their desks to face their computers and start putting together the plan on how we're going to pull this off. When we say a pivot, there was a literal pivot of chairs at the same time. There was a pivot of the company towards a new goal. In Sherlock's lab, on a recent day, scientist Paul Carson was tweaking the coronavirus test to make it faster and more sensitive. He's been working long hours with no complaints. So rarely in science do you get to work on something with such an immediate need, he says. When you're going through your PhD or other types of school, this motivation for work is always, well, maybe in 5 or 10 years or 20 years, this will be useful in developing a product. Basic research with longer-term goals does lead to breakthroughs, he says, but these days his work could already prove useful in a week or in a month. Sherlock coronavirus test got temporary federal approval called an emergency use authorization in May. Research associate Joel Mawat says CRISPR tests could help both the national shortage of tests and with concerns of testing accuracy. With the increase of the numbers recently, you can tell that some people don't believe that it's truly real, he said. So it's kind of cool to just put a little confidence back into the public eye. Sherlock Biosciences is partnering with a manufacturer to produce kits for testing in medical labs. An initial batch of 30,000, Donda says, that could scale up to millions if there's demand. It also just announced a deal for on-the-spot COVID test with a company whose chlamydia and gonorrhea test came back within about a half an hour, ideally while the patient is still in the office. There's competition, Trevor Martin, CEO of Mammoth Biosciences in South San Francisco, points to a recent paper Mammoth published on using its own test in patients, where we actually showed that Mammoth's CRISPR-based diagnostics are extremely effective tool for testing the COVID-19, he says. And anyone in diagnostics can tell you that a real patient samples are really the gold. Martin says Mammoth is working quickly as possible to bring their CRISPR test to the market. The way we think about it, it's really putting things out there that are effective and fill a gap in the market. And there's a lot of gaps in the market right now. The biggest gap, probably the biggest gap, is the need for quick, cheap, accurate coronavirus tests that can be done at home or at the entrances to a school or a nursing home or a stadium. MIT professor and Sherlock Biosciences co-founder Jim Collins predicts rapid progress on that front from a new technology called Inspector that would use synthetic biology methods to make a test on a simple paper strip. In a matter of small number of months, Collins says the company will be in a position to introduce an inspector-based test for COVID-19. Gutenberg and Abadiah have their own lab now at MIT's McGovern Institute and are working 7 days a week on a challenge of COVID testing. They're focused in part on developing a device akin to something as small as maybe a Coca-Cola can. Aboudier, I'm not probably saying that right, and that someone could do daily testing with every day for very low cost, maybe around $5 to $10. So they're also exploring various CRISPR enzymes, those scissors that do the molecular cutting, including one isolated from spoiled fruit juices. Because, nature's got a ton of amazing secrets in it, Gutenberg says. If there's a moral to this story, it's the importance of continuing to explore and try to understand the natural world. Yeah, so that they can rewrite it. It's important both because you never know when a basic science discovery like CRISPR could pan out, and because you never know when a natural phenomenon like the coronavirus could have huge and terrible impact on people's lives. Yeah. Let's see this first person treated with a CRISPR. This was in 2019. A powerful gene editing technique called CRISPR. But scientists have long hoped CRISPR, a technology that allows scientists to make very precise modifications to DNA, could eventually help cure many diseases. Like now, the coronavirus? For example, NPR has learned that a U.S. CRISPR study that's been approved for cancer at the University of Pennsylvania in Philadelphia has finally started. One patient had multiple myeloma, one had sarcoma. Both had relapsed after undergoing standard treatment. So the revelation comes as several other human trials of CRISPR are starting in U.S., Canada, and Europe. 2019 is the year when training wheels come off and the world gets to see what CRISPR can really do for the world in the most positive sense, says Fyodor Urnov, a gene editing scientist in the Altius Institute for Biomedical Sciences in Seattle and the University of California, Berkeley. Here are highlights of the year ahead in CRISPR research. So they tell us what CRISPR is. Let's see what their year ahead was since this was last year. Is this the same technique that caused a recent scandal when a scientist in China edited the genes of 2 human embryos? There's an important difference between the medical studies under discussion here and what the Chinese scientist did. He used CRISPR to edit genes in human embryos. That means the changes he made would be passed down for generations to come. And he did it before most scientists think it was safe to try. So what are they trying to say? That their edits they're going to make in us are not going to be passed down? I would beg to argue with that. So what's happening now with new or planned trials. We finally reached the moment when CRISPR is moving out of the lab. So let's see, almost all the studies have been in China. Now there's a clinical trial underway at the University of Pennsylvania using CRISPR for cancer treatment. It involves removing immune system cells from patients, genetically modifying them in the lab, and infusing the modified cells back into the body. The hope is the modified cells will target and destroy cancer cells. Wow. What other diseases are they testing treatments for? 2 trials sponsored by CRISPR Therapeutics in Cambridge, Mass, and Vertex Pharmaceuticals of Boston are designed to treat genetic blood disorders. One is for sickle cell disease and the other beta thalassemia. In fact, the first beta thalassemia patient was recently treated in Germany. First sickle cell patients could soon start getting the DNA in their blood. In this country, in Nashville, Tennessee, San Antonio, and New York. Lovely. Well, let's see. This study is noteworthy because it would be the first time scientists used CRISPR to edit genes while they're still inside the human body. The other studies involve removing cells from patients, editing the DNA, and then inserting them back into the patient's body. What can go wrong with CRISPR? Are there any concerns? Oh, no, of course not. Don't worry. Whenever scientists try something new and powerful, it always raises fears that something could go wrong. The early days of gene therapy were scarred by major setbacks, such as the case of Jesse Gelsinger, who died after an adverse reaction to a treatment. The big concern about CRISPR is that the editing can go awry, causing unintended changes in DNA that could cause health problems. Well, it seems they've gotten over their concerns with that because they're going to use the CRISPR technology in the new vaccine. There's also some concern about this new wave of studies because they're the first to get approved without going through an extra layer of scrutiny by the National Institutes of Health. That occurred because the NIH and the FDA changed their policy, saying only some studies would require an extra layer of review. Well, isn't that something? And now we're in 2020 and they were able to rush with even less scrutiny and rushed these things through. They came out with tests and now the vaccines. Well, CRISPR, they're just really profiting this year, aren't they? Every human on the planet should hope this technology works. But it might work. It might not. It's unknown, bioethicist at the University of Chicago said. This is an experiment. So you need exquisite layers of care, and you really need to think in advance with a careful ethical review of how you do this sort of work. Yeah, well, they seem to put that concern away. Wow, I just unbelievable. So, this in May, the FDA authorized the first ever CRISPR application for COVID testing. So, yeah, they're making quite a bit of money on these diagnostics. So let's see here. Nucleic acid detection with CRISPR nucleases. And then Sherlock, DETECTR and camera, 3 new CRISPR technologies. And this was in 2018. Jennifer, do you DNA? Where have we heard that name? Jennifer Doudna. Do you DNA? Jennifer does. She uses CRISPR. Sherlock uses CRISPR-associated protein known as Cas13, which binds to nucleic acid material. Researchers developed a version 2.0 of Sherlock that uses not 1 but 3 Cas13 enzymes and 1 Cas12a enzyme so that it senses multiple targets such as Dengue and Zika virus on a single reaction. To enhance Sherlock's capacity for field use, Zhang and his colleagues leveraged technology similar to pregnancy tests to create a paper test that displays results for a single genetic signature. Users dip the paper strip into a process sample and wait for a line to appear. The new paper readout for Sherlock lets you see whether your target was present in the sample without instrumentation. The Sherlock team also combined Cas13 with CRISPR enzyme CSM6 to increase the tool's sensitivity by threefold. In the study, they were able to find cancer mutations in cell-free DNA from saliva samples. All these improvements now give us even more diagnostic information and put us closer to a tool that can be deployed in real-world applications. Oh, kind of like 2020. Yeah. So, Dudna, a member of the departments of molecular and cell biology and chemistry at the University of California, Berkeley, the Howard Hughes Medical Institute, and Lawrence Berkeley National Lab, and her team discovered a new behavior about CRISPR enzyme CA12, which works similarly to CA9 in that it uses RNA as a guide to target specific DNA. Once it recognizes its target DNA, however, CA12 begins to cut single-stranded as well as double-stranded DNA. Because this single-stranded DNA threading activity is only unleashed after target binding, it can be used as an indicator of the presence of a certain target, Science editors explained. Researchers use this discovery to create a nucleic acid detection technology platform known as the DNA endonuclease targeted CRISPR transporter, or DETECTR. The platform combines Cas12a, its guide RNA, a fluorescent reporter molecule, and a recombinase polymerase amplification to create a single reaction. When warmed to body temperature, RPA rapidly multiplies the number of copies of the target DNA, boosting the chances that Cas12a will find one of them and bind and unleash single-stranded DNA cutting, resulting in a fluorescent readout. When applied to patient samples containing the human papillomavirus, DETECTR was able to successfully distinguish between HPV type 16 and 18. This illustrates how sensitive and precise the test is, its authors concluded. We want to push the limits of the technology, which is potentially applicable in a point-of-care diagnostic situation where there's a DNA component, including cancer and infectious disease. In a third study, researchers leveraged Cas9's target and slice abilities to develop two types of cell data recorders known as CRISPR-mediated analog multi-event recording apparatus, or camera systems. Get that. CRISPR-mediated analog multi-event recording apparatus, or camera systems. So their ability to what? See inside your DNA? So one of the approaches, camera, targets cells' plasmids, then monitors plasmids' ratios of live bacteria cells over time, recording the history of a live bacterial cell's exposure to nutrients, antibiotics, and other stimuli. Specifically, it records signals by changing the ratio of two DNA plasmids, A and B, in bacterial cells, using Cas9 and a guide RNA to selectively target and destroy just one of the plasmids. In the event A is destroyed, the result is the cell starts to replicate both A and B to fill in the loss of A. As a result, the ratio of AB decreases over time. The more intense the signal, the longer the signal, the lower the ratio drops. Another approach, camera 2, records signals by linking a signal of interest to the production of a base editor, which in turn records the signal directly as a change in the DNA sequence at a specified position in the genome or in plasmid DNA. It works in both bacteria and mammalian cells. His team said he was able to demonstrate the ability of camera 1 and 2 to record a cell's exposure to nutrients, antibiotics, light, viruses, and even a kinase inhibitor that changes WNT signaling in human cells. His and other labs are in the process of using the camera system to study cell signaling during differentiation and other events marked by dramatic change in cell states. So there you go. There's the camera, so they can see as they edit your genes and rewrite it. I don't know. This is just so disturbing. Sherlock, DETECTR, and camera. There you have it. And George Church said everything involved in the COVID involves synthetic biology and CRISPR. the testing, the vaccines, the therapeutics, all of it. Thanks for listening, guys.